Welcome to the Devonian Period, the age of fishes, a chapter of Earth's history so fundamental, so foundational that without it, nothing you see swimming, walking, or breathing air today would exist. This is the story of how life in the sea grew bold, grew armored, grew ambitious, and ultimately grew legs. Please let us know in the comments where you are watching this history video and what time you are watching it, and be sure to subscribe to the channel for future videos. Imagine standing at the edge of a sea that no human eye has ever seen. The sky above you carries a familiar blue, but the sun burns slightly brighter, sitting in an atmosphere still finding its oxygen balance. The land behind you is mostly bare, raw rock, thin crusts of early soil, and scattered mats of primitive plants clinging to riverbeds and coastlines like green whispers against an otherwise brown and barren world. But the water in front of you, that is where everything is happening. That is where written, one creature at a time, through hundreds of millions of years of patient, relentless experimentation. The Devonian period stretches across an enormous sweep of geological time, from approximately 419 million years ago to around 359 million years ago, a span of roughly 60 million years during which the oceans of Earth transformed from relatively simple communities of ancient marine life into complex, layered ecosystems bursting with competition, predation, and innovation. 60 million years. To put that in perspective, 60 million years ago, dinosaurs had just gone extinct, and the first primitive primates were only beginning to feel their way through a recovering world. That is the same length of time the Devonian lasted, a full dinosaur era's worth of evolution, compressed into a chapter that most people have never heard of. Yet this period deserves to be known. It deserves to be understood, because what happened in those ancient seas was not simply a matter of fish swimming around. What happened was the invention of the vertebrate body plan, the development of jaws, the construction of internal skeletons from living bone rather than simple cartilage, the birth of paired fins that would, given time and pressure, eventually reshape themselves into arms and legs. The Devonian was not just an era of fish, it was the era in which fish became something more than fish. It was the era in which the ancestors of every backbone creature on Earth, every reptile, every mammal, every bird, and every human being were hammered into shape by the forces of competition, survival, and change. To understand the age of fishes, we have to begin even earlier, not in the Devonian itself, but in the long, slow build-up that preceded it. Life on Earth had existed for an extraordinary length of time before the Devonian arrived. Microbial life had colonized the oceans as far back as 3.5 billion years ago. Simple multicellular organisms had appeared and diversified in the late Precambrian. The Cambrian explosion, roughly 538 million years ago, had produced an astonishing burst of animal diversity, strange, soft-bodied, and armored creatures filling the seas with a variety of body plans that remains one of the great wonders of paleontology. By the time the Ordovician period followed, the Cambrian marine ecosystems had grown significantly more complex, with corals, mollusks, echinoderms, and early vertebrates beginning to carve out ecological roles in a world of shallow tropical seas. But the vertebrates of that early era, the Ordovician and the Silurian periods that followed, were humble creatures by any measure. They were jawless, they were small. Many of them filtered food from sediment or water, living out quiet lives along the ocean floor or in the mid-water column, consuming whatever nutrients drifted within reach. They looked in many ways more like oversized tadpoles or flattened worms than anything we might recognize as a fish today. Yet they carried within their bodies the fundamental architecture that would one day produce the entire vertebrate lineage, a notochord, a dorsal nerve cord, and the beginnings of a skull and brain case that would, in time, become the foundation of all animal intelligence. The Silurian period, which immediately preceded the Devonian, was a time of gradual build-up. The great extinction event that had closed the Ordovician, one of the most severe in Earth's history, triggered by a rapid glaciation that drained sea levels and froze much of the planet's shallow marine habitat, had swept away vast numbers of species, resetting ecosystems and leaving behind a world of survivors ready to diversify into emptied niches. The Silurians saw the recovery and expansion of reef-building communities, the spread of early vascular plants onto land, and the continued diversification of jawless fish alongside the first stirrings of jawed vertebrates. By the end of the Silurian, the stage was set, the world was primed, and then the Devonian began and everything accelerated. The Earth of the early Devonian was a planet arranged very differently from the world we know. The continents were positioned in unfamiliar configurations. The great southern supercontinent of Gondwana dominated the southern hemisphere, a massive, slow-drifting landmass that held within it the future territories of Africa, South America, Antarctica, Australia, and India. To the north, the smaller landmasses of Laurasia, combining what would eventually become North America, Europe, and parts of Asia, were clustered together near the equator, bathed in warm, shallow tropical waters that would prove extraordinarily productive for marine life. Between and around these landmasses, ancient seas spread in patterns that have no modern equivalent: the Rake Ocean, the Tethys Sea, vast shallow epicontinental seas covering the low-lying margins of continental shelves with warm, sunlit water that was, in terms of biological potential, possibly rich. The climate of the Devonian was, for much of the period, warm and relatively humid. There were no polar ice caps of significant size in the early and middle Devonian. The planet ran equatorial temperatures in the ocean, potentially reaching levels that would be considered by today's standards, and with tropical marine environments particularly abundant in the shallow water zones along continental margins. This warmth was biologically important. Warm water holds less dissolved oxygen than and tabulate corals that formed the underwater architecture of Devonian marine communities. These reefs were not identical to modern coral reefs, but they served the same ecological function, providing structure, shelter, and surface area that allowed communities of organisms to layer themselves in 3 dimensions, creating a complexity of habitat that drove the diversification of everything that lived there. It is in this context, warm seas, productive shelves, complex reef systems, and the ecological inheritance of Silurian survivors, that the age of fishes truly begins. And it begins not with a single dramatic event, but with a slow, magnificent expansion of possibility, as creatures that had been testing new designs for millions of years began, finally, to find designs that worked extraordinarily well. The first thing to understand about Devonian fish is that the word fish does not begin to capture what they were. We use the term loosely today, grouping together an enormously diverse array of aquatic vertebrates under a single kill. But the Devonian was an era in which the concept of fish was itself being invented, in which the boundaries between different vertebrates were still being negotiated by evolution, and in which creatures existed that do not fit neatly into any category we recognize from the modern ocean. The dominant vertebrates of the early Devonian were the placoderms, a group whose name means plate-skinned in Greek, and whose appearance justifies fully. Placoderms were armored fish, or more precisely, armored vertebrates, because calling them simply fish understates how different they were from anything swimming in today's oceans. Their heads and thoraxes, the front portions of their bodies, were encased in thick, interlocking plates of bone and dentine, forming a rigid external shield that covered their organs and gave them the appearance of creatures wearing medieval armor. This armor was not decorative. It was functional, protective, and in many cases extraordinarily effective. The placoderms were, for the majority of the Devonian period, the most successful and diverse group of jawed vertebrates on Earth, occupying every marine habitat from open ocean to shallow reef to freshwater river and lake. But the armor tells only the innovation that placoderms carried, the innovation that would echo through the entire subsequent history of life, was the jaw. Jaws, the simple, seemingly obvious mechanical apparatus that allows an animal to bite, to grip, to crush, and to actively seize food from its environment rather than simply filtering or absorbing it. Before Jaws, vertebrate life was fundamentally passive in its relationship with food. Jawless fish, the agnathans, could consume only what they could filter, rasp, or suck into their mouths. They were dependent on soft food sources, on sediment-dwelling organisms, on the slow gathering of nutrients from water or substrate. They could not bite, they could not pursue and capture prey in the way that a predator requires. The evolution of Jaws changed all of that in a single stroke of biological innovation. And it is worth pausing to understand how this transformation happened, because it represents one of the most remarkable examples of evolutionary repurposing in the entire history of life. Jaws did not appear from nowhere. They were not invented fresh. Instead, they were built from existing structures, specific gill arches that supported the breathing apparatus of early jawless fish. Gill arches are curved bars of cartilage or bone that support the gill tissue through which fish extract oxygen from water. In jawless fish, a series of these arches curved around the throat and pharynx, holding the gills in place and allowing water to flow over them efficiently. Over time, over the slow accumulation of tiny genetic changes across many generations, the frontmost pair of these gill arches began to shift. They moved forward, they changed shape, they became hinged at the midpoint. And gradually, across millions of years of transition that we can track, however imperfectly in the fossil record, they transformed into the upper and lower components of a jaw, a structure that could open and close, that could apply force, that could bite. This transformation, the jaw, is one of the pivotal moments in the history of vertebrate life. It opened up ecological possibilities that had been entirely unavailable before. An animal with jaws could eat things that were hard, could crack shells, crush bones, bite through tough vegetation. It could actively pursue prey, clamping down rather than simply filtering. It could manipulate food items, holding them while tearing them apart. It could, in short, become a predator in the full modern sense of that word, a creature that actively hunts and kills, that exerts force and intent upon other organisms in the pursuit of nutrition. The placoderms were among the earliest jawed vertebrates to diversify extensively, and their success across the Devonian speaks to how powerful this innovation was. But they were not alone. Alongside them, other groups of jawed fish were also developing and diversifying, the acanthodians, sometimes called spiny sharks for their sharp fin spines, though they were not true sharks. The early chondrichthyans, the cartilaginous fish that would eventually give rise to modern sharks and rays, and the osteichthyans, the bony fish whose two great lineages, the ray-finned fish and the lobe-finned fish, would between them come to dominate every aquatic environment on Earth and eventually colonize the land. Let us spend time with the placoderms first, because they deserve it. These were the rulers of the Devonian seas, and their story is one of the most fascinating chapters in the history of life, a story of extraordinary success followed by equally extraordinary extinction, of innovation right up until the moment the world changed and they could not change with it. The variety within the placoderms was staggering. The group included species ranging from a few centimeters in length to true giants that rank among the largest predators of any era. It included flat-bodied bottom dwellers that glided along the seafloor like living tanks, crushing shellfish and invertebrates between their bony dental plates. It included streamlined, open-water hunter prey through the melon with bead and a filter through plankton with mouths held wide open. And it included, most famously, the apex predators, the great armored hunters that sat at the top of Devonian food chains and defined the ecological character of the period's seas. Among these apex predators, one genus stands out above all others as a symbol of Devonian life, Dunkleosteus. The name comes from David Dunkel, a paleontologist who worked extensively with Devonian fossils, and it is a name that has become nearly synonymous with the concept of ancient marine predation. Dunkleosteus was immense. Current estimates of its size vary, and paleontologists, but the largest specimens suggest an animal that may have reached lengths of 6 to 9, roughly the size of a modern great white shark, with a head encased in armor so thick and heavy that the bony shield alone resembled feet of biological. But Dunkleosteus was not simply large. The name comes from David Dunkel, a paleontologist who worked extensively with Devonian fossils, and it is a name that has become nearly synonymous with the concept of ancient marine predation. Dunkleosteus was immense. Current estimates of its size vary, and paleontologists debate the details, but the largest specimens suggest an animal that may have reached lengths of 6 to 9 meters, roughly the size of a modern great white shark, with a head encased in armor so thick and heavy that the bony shield alone represents a remarkable feat of biological engineering. But Dunkleosteus was not simply large. It was powerful in a way that goes beyond mere size. The jaw mechanism of this animal was unlike anything in the modern ocean. A system of interlocking bone plates allowed the mouth extraordinary speed and force, creating a rapid suction that drew prey inward while the jaws simultaneously slammed shut with a bite force estimated to be among the highest of any fish that has ever lived. Studies of the biomechanics of Dunkleosteus skulls have suggested bite forces in the range of several thousand newtons, enough to crush the armor of other placoderms, enough to bite through the shells of large invertebrates, enough to make this animal the unchallenged apex predator of the seas it inhabited. What makes Dunkleosteus even more remarkable is the design of its teeth. Placoderms did not have teeth in the conventional sense. They did not have the individual, separately rooted tooth structures that we find in sharks and bony fish. Instead, they had bony plates that formed the margins of their jaws, plates that were self-sharpening, in a sense, because the upper and lower plates wore against each other during biting, maintaining sharp edges through constant use. This was an elegant solution to the problem of tooth, where that avoided the metabolic cost of continuously producing new tooth structures, and it worked. It worked for millions of years, across the enormous diversity of placoderm species that populated Devonian waters. The armor of Dunkleosteus and its relatives raises its own set of fascinating questions. Why would an apex predator, a creature with no significant natural enemies, need heavy armor? Part of the answer lies in developmental history. The placoderms inherited their armored body plan from ancestors who very much did need protection, and the armor was already established as a fundamental aspect of placoderm anatomy before these animals reached the apex of their ecological range. But there is also evidence that the armor served functions beyond simple defense. It may have played roles in display, in species recognition, in the regulation of buoyancy, and even in the structural support of muscles that powered the jaw mechanism. The armor was not simply a passive shield, it was an integrated system that worked with the animal's entire physiology. Beyond Dunkleosteus, the placoderm world was populated by a remarkable cast of other armored creatures, each adapted to its own ecological niche with the precision that evolution, given enough time, invariably achieves. Consider Bothriopes, a placoderm that was among the most widespread vertebrates of the Devonian, found in fossil deposits on virtually every continent, suggesting a cosmopolitan distribution across both marine and freshwater environments that speaks to its extraordinary ecological flexibility. Bothriopes was not a predator in the Dunkleosteus mold. It was smaller, more modestly proportioned, with a distinctive pair of long, jointed pectoral appendages, armored, fin-like structures that extended from the sides of its thoracic shield like arms, used to push along the bottom, to prop the animal up and possibly to minimize or dig into sediment in search of invertebrate prey. The pectoral appendages of Bothriopes are particularly interesting from an evolutionary perspective, because they represent one of several independent experiments in early vertebrate history with the idea of paired, limb-like structures. The pectoral fins of fish are homologous to the forelimbs of land vertebrates. They are, in a fundamental developmental sense, the same, expressed in different ways by different lineages. When we see Bothriolpes pushing itself along the seafloor with jointed, articulated appendages, we are seeing a distant, heavily armored cousin of the same body plan and innovation that would eventually, in the lobe finnage, produce the first truly limbs and pave the way for the colonization of land. Another remark worth examining in detail is a giant of a different Dunkleosteus, and in some ways an even more surprising one. Titanishes was enormous, comparable in size to Dunkleosteus by some estimates, but its jaw structure tells a completely different ecological story. Where Dunkleosteus had powerful, shearing dental plates suited for crushing and biting hard prey, Titanishes had reduced, almost vestigial jaw structures that appear to have been poorly suited for capturing and processing large prey items. The most widely accepted interpretation is that it was a filter feeder, an animal that swam through plankton-rich water with its enormous mouth agape, small organisms in a manner analogous to modern basking sharks or whale sharks. In the Devonian seas, even the giants had diverse ecological strategies. Titanishes reminds us that large body size is not always about predation. It can equally be a strategy for exploiting the sheer abundance of microscopic life that saturates productive marine ecosystems. The placoderms also occupied freshwater environments with considerable success. Freshwater fish communities of the Devonian are documented in remarkable fossil beds around the world, in particular, in the red sandstone deposits of what is now Scotland and in the Gogo Formation of what is now Australia, and these deposits reveal rich, complex communities of placoderms living alongside early sharks, lobe-finned fish, and ray-finned fish in lakes, rivers, and estuaries. The freshwater placoderms show adaptations to their specific environments. Some were shallow water bottom dwellers, some were mid-water hunters, and some appear to have been capable of moving between freshwater and marine environments, a physiological flexibility that would have been enormously advantageous in a world where sea levels fluctuated and river deltas created complex gradients between salt and fresh water. It is at this point, with placoderms established as the dominant jawed vertebrates of the Devonian, that we must introduce the other major players, the groups that were less dominant during the Devonian itself, but would ultimately inherit the Earth's waters after the placoderms disappeared. These are the chondrichthyans, the cartilaginous fish, ancestors of modern sharks and rays, and the osteichthyans, the bony fish, whose descendants today represent the overwhelming majority of all species. The early sharks of the Devonian were very different from the sleek, powerful and from modern oceans. They were smaller, more varied in form, and experimentally diverse in ways that reflect the general character of Devonian vertebration, a period in which many different designs were being tested silently, with no guarantee of which would prove most successful in the long run. The earliest well-documented sharks appear in the Devonian fossil record as isolated scales and fin spines rather than as complete skeletons, because shark skeletons are made of cartilage rather than bone and therefore do not preserve as readily in the fossil record. But from these fragmentary remains, and from a handful of more complete specimens that have been found in exceptional preservation, we construct what early sharks and how they lived. One of the well-known sharks is Dunkleosteus, found primarily in the late Devonian deposits of what was once an inland sea. Dunkleosteus was a slender, streamlined animal, reaching perhaps 1.5 meters in length, with large eyes, a terminal mouth, positioned at the very front of the snout rather than on the underside, as in many modern sharks, and a symmetrical, forked tail that suggests it was an active, fast. Unlike many later sharks, Cladoslash appears to have lacked the claspers that male sharks use for internal fertilization in modern species, suggesting that its reproductive biology, modern chondrichthyans, perhaps more similar to the external fertilization practiced by many bony fish. The significance of early sharks in the Devonian goes beyond their own biology, however. They represent the early expression of a body plan, cartilaginous skeleton, streamlined form, continuously replaced teeth, active predatory lifestyle that has proven to be one of the most durable in the history of vertebrate life. Sharks are sometimes described as living fossils, a term that paleontologists use cautiously because it implies that these animals have not changed when in fact they have evolved continuously. But the basic shark design, efficient, flexible, built around a cartilaginous skeleton that is lighter than bone and allows remarkable agility, has proven adaptable enough to persist through multiple mass extinctions and across radically different ocean conditions. The Devonian sharks were exploring that design in its earliest phases, finding its parameters, testing its limits, and beginning the long process of eventually produce the diverse and the fauna of later periods. The cartilaginous skeleton that defines sharks and rays deserves special attention because it represents a fundamentally different approach to building a vertebrate body placoderms and bony fish were pursuing simultaneously. Cartilage is lighter than bone, more flexible, and less metabolically expensive to produce and maintain. A skeleton made allows an animal to grow quickly, water, reducing the energy cost of maintaining, and to flex and twist. Bony skeleton does not easily. These advantages come with cartilage does not preserve as well as bone. It cannot support the same mechanical loads as dense bone in some, and it provides a different set of attachments. But for a swimmer whose primary medium of movement, cartilage offers genuine advantage. Kept the chondrichthyan design for hundreds of millions of. Now we must turn to the bone, as it is within this group that the most consequential developments of the Devonian period occurred. Very early in their history into two great lineages, the actinopterygians or ray-finned fish and the sarcopterygians or lobe-finned fish. Today, the ray-finned fish are the dominant vertebrates of aquatic environments, comprising the vast majority of all living fish species: salmon, tuna, cod, bass, perch, and nearly every other fish familiar from rivers, lakes, and oceans. But during the Devonian, it was the lobe-finned fish that were the more prominent and ecologically significant group, and it was from within the lobe-finned fish that one of the most important transitions in the history of life would eventually emerge: the move from water to land. The difference between ray-finned and lobe-finned fish is fundamental and immediately visible in their fin structure. Ray-finned fish have fins that are supported by thin, bony rays, slender projections of bone, arranged in fans that provide surface area and maneuvering. These fins are effective swimming appendages, but they are lightweight structures with limited capacity for bearing weight or against gravity on a solid surface. Lobe-finned fish, by contrast, have fins that are supported by a fleshy lobe, a thick, muscular base that contains an internal skeleton of bones arranged in a pattern recognizable as a precursor to the limb bones of land vertebrates. The pectoral and pelvic fins of lobe-finned fish contain bones that correspond developmental terms to the humerus and femur, to the radius and ulna and tibia and fibula, and eventually to the bones of wrists and ankles and fingers. These are not metaphorical correspondences, they are real, traceable homologies, documented by the genes that control limb development and confirmed by the fossil record of transition. In the Devonian seas, lobe-finned fish were diverse and ecologically important. Among the most significant is Eusthenopterus, a lobe-finned fish from the late Devonian of what is now Canada and Europe that has become one of the most studied animals in the entire fossil record precisely because of what its anatomy reveals about the transition between aquatic and terrestrial life. Eusthenopteran was a fairly large fish, perhaps a meter in length, with a powerful muscular body, large pectoral and pelvic fins with well-developed internal bone structure, and anatomical features of its skull and braincase that show clear relationships to early land vertebrates. It was not itself making the transition to land, it was a fully aquatic animal, but it documents the kind of body plan from which the first terrestrial vertebrates would emerge. The internal skeleton of Eusthenopteran's pectoral fin shows a single proximal bone, the equivalent of the humerus, attached to the shoulder girdle, followed by two bones arranged side by side, the equivalents of the radius and ulna, and then a series of smaller bones beyond. This pattern, one bone, then two bones, then several smaller bones, is the exact pattern found in the forelimbs of all tetrapods, the four-limbed vertebrates that colonize the land. Seeing it in a Devonian fish is one of the most striking demonstrations available to us of how evolution works through gradual modification of existing structures, reshaping what already exists rather than inventing new things. Another lobe-finned fish of the Devonian that deserves mention is Panderichthys, a flattened, somewhat crocodile-like animal from the late Devonian of Latvia that shows even more tetrapod-like features than Eusthenopteran. Panderichthys had a flat head, eyes positioned on the skull rather than on the sides, and pectoral fins with an even more limb-like internal skeleton. It was apparently adapted for life in very shallow water, perhaps moving through aquatic vegetation in marginal, swampy environments where the ability to prop itself up and push against the substrate would have been genuinely useful. Panderichthys sits even closer to the transition between fish and land vertebrates than Eusthenopteran does, and its existence in the fossil record documents how that transition was not a sudden leap, but a gradual accumulation of anatomical changes across a series of forms, each more terrestrially capable than the last. The ecological context of the Devonian seas was not simply a backdrop against which these vertebrate dramas played out, it was an active participant, shaping the evolution of every creature that lived within it. The reef communities of the Devonian were among the most productive marine ecosystems of any geological period, and understanding them is essential to understanding why vertebrate evolution proceeded so rapidly and during this time. Devonian reefs by stromatopteroids, sponge-like organisms that secreted calcium carbonate skeletons in layered, dome-shaped or branching colonies, and by tabulate and rugose corals, ancient relatives of modern corals that constructed colonial or solitary skeletal structures of their own. These reef builders created the same kind of complex, three-dimensional habitat that modern coral reefs provide, a maze of crevices, overhangs, walls, and open spaces that offered shelter for small organisms, hunting grounds for larger ones, and attachment surfaces for a diverse community of filter feeders, grazers, and scavengers. The reef provided structure, and structure enabled diversity. Around and within these reefs lived communities of enormous complexity. Brachiopods, clam-like filter feeders that were among the most abundant shellfish of the Paleozoic, covered hard surfaces in dense aggregations. Crinoids, beautiful, flower-like animals related to sea stars, waved their feathery arms in the current from attached stalks, catching food particles drifting past. Trilobites, the iconic three-lobed arthropods, the most recognizable symbol of ancient marine life, still crawled across the seafloor in the Devonian, though their diversity was declining from the heights they had reached in earlier periods. Ammonoids, coiled cephalopods related to the modern nautilus, but diverse far beyond anything that survives today, swam through the water column in great variety, predators and prey both, their beautiful shells preserving in the fossil record the record of countless infants. And through all of this, over the reefs, among the sponges, in the open water above the shelf, along the muddy seafloor at the reef's base, the fish moved. All of them. The armored placoderms picking their way along the bottom, their bony shields glinting in filtered light. The early sharks slicing above the reef, hunting for smaller fish and invertebrate. Finned fish lurking in ambush among vegetation in shallower zones, waiting for prey to come within range of a sudden lunge. The ray-finned fish darting in tight schools through corridors of reef structure, their mirror like scales catching and scattering light. The jawless fish, surviving remnants of a pre-jaw world, smaller and less ecologically dominant than their jawed relatives but still present, still finding niches in the complex mosaic of Devonian marine life. The diversity of feeding strategies that evolved in this context was remarkable. Every possible approach to nutrition that an aquatic animal might pursue was being explored simultaneously by different lineages in different ways. Active predation, pursue, capture, kill, consume, was pursued by placoderms like Dunkleosteus and early sharks alike. Filter feeding, swim or station feed, collecting small particles from the water, was practiced by titanischthys and by various invertebrate components of the community. Bottom feeding, root through sediment, crush shells, excavate burrowing vertebrates, was the strategy of placoderms and various other bottom dwellers. Ambush predation, lie still and strike fast, was the approach of the lobe-feeding in vegetation. And grazing, nibbling at algae or sessile invertebrates, was practiced by yet another suite of smaller, less charismatic but ecologically essential species. This ecological complexity, the layering of different species into different niches, each one finding its own way to extract energy from the productive Devonian Sea, created a web of interactions that was, by any measure, sophisticated. Predator-prey relationships drove evolutionary arms races. Prey species evolved better camouflage, faster swimming, harder shells. Predators evolved more powerful bites, better sensory systems, greater speed or stealth. Competition for food and space drove specialization. Species that were too similar in their ecological roles either divided their habitats or drove each other toward extinction. The reef provided the structure that made all of this possible, the habitat complexity that allowed many species to coexist in a relatively small area by partitioning space and resources among themselves. The sensory systems that Devonian fish developed in response to these ecological pressures deserve attention because they represent innovation with modification in the sensory fish and other aquatic vertebrates today. The challenges of navigating, hunting, and avoiding predation in a complex three-dimensional drove the elaboration of sensory systems far beyond what the earliest vertebrates had possessed. Vision was important. The eyes of many Devonian fish were large relative to their body size, an indicator across the animal kingdom that vision is a primary sensory modality for a given species. Large eyes collect more light, allowing detection of objects in dimmer conditions and at greater distances. In the turbid, potentially reduced light conditions of reef environments or deeper water, visual acuity was a survival advantage. See its prey before the prey could see it had an enormous competitive advantage. The forward-positioned eyes of some lobe-finned fish, like pandurich eyes on top of the head, may have allowed these animals to watch for terrestrial or aerial threats while remaining mostly submerged in shallow water, an early hint of the visions that would be important for early land vertebrates. The lateral line system, a sensory organ unique to fish and aquatic amphibians, was also well developed in Devonian vertebrates. The line consists of a series of sensory receptors distributed along the sides of the body and in channels across the head, capable of detecting pressure changes and water movements in the surrounding environment. This allows a fish to sense the movements of other animals, the bow wave of an approaching predator, the turbulence created by a school of prey fish, the pressure changes associated with obstacles without using all. Mind is effectively no direct human equivalent, a whole body awareness of mechanical disturbances in the surrounding fluid that provides information about the environment that vision alone cannot. In the murky darkness among the complex microhabitats of the reef, the lateral line was invaluable. Predators could track prey by the pressure waves their movement created. Schooling fish could sense the movements of their maintained formation without visual contact. Animals could navigate around obstacles in low light. The lateral line made it possible to be effective in conditions where vision was limited, and its presence in Devonian fish, documented by the sensory pore systems preserved in fossil skulls, confirms that these ancient creatures were operating with sensory sophistication that went well beyond simple light detection. The sense of smell, chemoreception, was also important in virtually all fish today. The nasal capsules of Devonian fish were well-developed structures, and the brain regions associated with olfactory processing were proportionally large in many species, suggesting that chemical detection of food, mates, and environmental conditions was a primary sensory modality. For predators hunting in open water where visual contact with prey was limited, the ability to follow chemical trails, gradients of dissolved by potential prey, provided a way to locate food sources across distances that vision could not span. For prey species, the chemical detection of predator-associated compounds could trigger avoidance behavior that saved lives. Chemistry was the language of the Devonian Sea, and every fish was fluent in it to some degree. The reproductive biology of Devonian fish is another area where remarkable discoveries have illuminated these ancient creatures. For a long time, fish reproduction was assumed to follow the simplest possible pattern: external fertilization, with eggs and sperm released into the water simultaneously and left to develop without parental care. This pattern is common among modern fish and requires minimal physiological complexity. But the fossil record of Devonian placoderms has revealed something far more surprising: evidence of internal fertilization and viviparity, live birth, in fish that lived more than 350 million years ago. The discovery came from specimens of a placoderm called Matterpiscus attenboroughi, a name that honors the natural attenborough, found in the Gogo Formation of Australia, one of the world's most important sites for three-dimensional preservation of Devonian vertebrate fossils. Inside the body cavity of one Matterpiscus specimen, paleontologists found the remains of a juvenile individual connected by a structure interpreted in the arms reaching for this narration, in the legs carrying it forward, in the hands that hold and the lungs that breathe. All of it traces back ultimately to the shallow, sunlit, complex, and dangerous seas of the Devonian period, the age of fishes, the foundational chapter of vertebrate history. The story of this ancient world is far from finished, even in the telling. We have introduced the major players, established the ecological context, traced the rise of jaws and armor and bony skeletons and lobe fins. But there is much more to explore: the specific dramas of individual species and communities, the mechanisms of the late Devonian extinction events, the fate of the various lineages that survived or perished, and the detailed those survivors carried the Devonian into the world that came after. The second part of this documentary will take up all of these threads, carrying the story through the great ecological crisis of the late Devonian and into the dawn of the world that followed, a world shaped entirely by what the Devonian had built and what it had destroyed. But for now, let us stand for a moment at the midpoint of this ancient history and allow the weight of it to settle. 400 million years and more separate us from these creatures. The mountains that rose above their seeded and replaced, the continents that have drifted to entirely different positions, The atmosphere they breathed and the in have been cycled through countless living bodies since theirs. Nothing of the specific physical Devonian world survives above ground. It persists, the pattern that evolution laid down in those ancient waters, the vertebrate body plans and its internal skeleton and its paired appendages survives, modified but recognizable, in every fish and frog and lizard and bird and mammal alive today. The Devonian did not simply happen, it built the future. Every vertebrate alive today, every single one without exception, is a descendant of creatures that swam in Devonian seas or in the transitional margins where those seas met the land. The Age of Fishes was not a prologue to the real story of vertebrate life. It was the first chapter of that story, the chapter in which the characters were introduced, the themes were established, and the foundation was laid for everything that followed. Understanding it is not merely an act of historical curiosity. It is an act of self-knowledge, the discovery of where we came from, how deeply our roots go, and how ancient is the heritage that we carry in our very bones. The fossils that preserve this ancient world are themselves remarkable objects, not just for the information they contain, but for the improbability of their existence. The conditions required to produce a well-preserved fossil are stringent and rarely met. An organism must die rapidly in sediment before its remains are scattered by scavengers or dissolved by decomposition. The sediment must be fine-grained enough to preserve anatomical detail. The buried remains must escape dissolution by groundwater, deeply buried or destroy their structure, uplift and erosion that eventually brings this, and then must be found by the right human at the right time before continued erosion destroys them. At every step, the odds are against preservation. The fact that we have any fossils at all is something of a miracle. The fact that we have fossils as complete and detailed as some Devonian specimens, three-dimensionally preserved fish from the Gogo Formation, articulated placoderm shields from the Cleveland Shale, the extraordinary sequence of lobe-finned fish documenting the transition to tetrapods, is almost extraordinary beyond comprehension. Yet they exist. These windows into an ancient world, these improbable accidents of geology and chemistry and human curiosity, exist and have been found and studied and interpreted by generations of paleontologists who dedicated their lives to understanding them. And through their work, through the slow, patient, meticulous examination of stone and bone, of chemical signature detail, we have reconstructed this vanished world with a level of confidence and precision that would have been unimaginable a century ago. The Devonian seas live again in our understanding, in our ability to visualize them, to populate them with their extraordinary inhabitants, to trace the connections between those ancient creatures and the living world around us. That is the gift of paleontology, the gift of deep time, recovered from the rock and handed to the present. And it is a gift that the Age of Fishes offers in particularly generous measure, because the Devonian was so consequential, so creative, so full of invention and diversity and drama, that it rewards examination at every level of detail. From the mechanics of the placoderm jaw to the chemical signaling of reef communities, to the first tentative terrestrial experiments of lobe-finned fish in shallow marginal waters, every aspect of Devonian life reveals something important about how the living world works, how it changes, how it builds on what came before and prepares the ground for what comes after. The Age of Fishes is not an obscure footnote in the history of life. It is one of its defining chapters, a time when the vertebrate lineage found its design and began the long journey that would eventually bring it here, to this moment, to this telling, and to every living thing that draws breath and moves through the world on the inheritance of ancient fins. We left the Devonian world at a moment of extraordinary richness, seas crowded with armored giants and sleek early sharks, reefs humming with biological complexity, and along the margins of ancient waterways, the first tentative experiments of lobe-finned fish testing the boundary between water and land. The world was full, it was productive, It was, by any measure of biological success, thriving. And yet beneath that apparent abundance, forces were gathering that would, over millions of years, dismantle the very ecosystems that had made the age of fishes so remarkable. Part 2 of our journey into the Devonian takes us deeper into that complex story, into the lives of creatures we have not yet met, into the specific dramas of ancient communities, into the mechanisms of environmental change, and ultimately into the great crisis that closed the Devonian and reshaped the trajectory of vertebrate life forever. The age of fishes was not simply an age of flourishing. It was also an age of reckoning, a period in which the innovations of evolution were tested not just against each other, but against a planet that was itself in motion, changing in ways that no living thing could anticipate or prepare for. This is the rest of that story. To fully appreciate what was at stake in the late Devonian, we must first deepen our understanding of the ecological web that had been constructed during the period's long middle phase, the intricate, interdependent community of organisms that filled the ancient seas and gave them their character. We have discussed the dominant vertebrates, the placoderms, the early sharks, the lobe-finned and ray-finned bony fish, but the ocean is never simply a collection of fish. It is a system, a network of relationships between organisms at every level of biological organization, from the microscopic photosynthetic plankton at the base of the food web to the apex predators at its summit. And the non-vertebrate components of the Devonian marine community were, in their own right, as remarkable and as ecologically important as the fish that swam among them. Consider the invertebrate world of the Devonian Sea. The reef communities that we have already described were populated not just by the coral and stromatoporoid builders, but by an enormous diversity of organisms that lived on, in, and around the reef structure. Brachiopods, those filter-feeding shellfish that dominated Paleozoic seafloors in the way that clams and oysters dominate modern ones, were present in astonishing abundance and variety. Hundreds of brachiopod species are known from Devonian deposits around the world, each one adapted to its own specific microhabitat within the larger reef community, some living on hard surfaces in high-current areas, catching food from the rushing water, some nestled in crevices where current was minimal, some partially buried in soft sediment at the reef base, filtering the turbid water that settled there. The brachiopods were the grazers and filter feeders of the reef community's lower ecological tiers, converting planktonic production into animal biomass that supported the larger creatures above them in the food web. Alongside the brachiopods, the echinoderms contributed enormously to Devonian reef ecology. Crinoids, the sea lilies, were among the most beautiful and most ecologically important animals of the Devonian seas. These animals, related to ours and sea urchins, but superficially resembling flowers, anchored themselves to hard surfaces by means of a long, flexible stalk and extended elaborate, feathery arms into the current to collect food particles. In life, they would have transformed the surfaces of Devonian reefs into waving, colorful gardens of feeding arms, dense aggregations of crinoids covering every available hard surface, collectively filtering enormous volumes of water and converting dissolved organic material into animal tissue. When crinoids died, their skeletal debris, calcite discs and arm segments, accumulated in thick layers around the reef, forming a significant component of the carbonate sediment that eventually became the limestone we quarry today and find fossil fish within. Trilobites, those iconic three-lobed arthropods that had been a defining feature of marine ecosystems since the Cambrian, were still present in the Devonian, though their diversity had declined from its Cambrian and Ordovician peaks, and they would not survive into the Permian. Devonian trilobites were diverse enough in their own right, occupying a range of ecological roles from benthic scavengers and predators to mid-water swimmers. Some Devonian trilobites had evolved remarkable anatomical specializations, greatly enlarged eyes for vision in dimly lit environments, smooth, streamlined bodies for active swimming, or elaborate spiny exoskeletons that made them more difficult to swallow. The trilobites of the Devonian were animals that had been refined by hundreds of millions of years of evolution, and they showed it in the precision of their adaptations. But they were animals in decline, their ecological dominance being usurped by a more modern caste of invertebrates, and the environmental stresses of the late Devonian would hit them hard. The ammonoids, cephalopods with coiled shells, related to the modern nautilus but far more diverse, were ecological innovators of the Devonian seas. Unlike the nautilus, which is a slow-moving, deep-water scavenger, many Devonian ammonoids were active, fast-moving predators of the open water, using their jet propulsion system and their tentacles to pursue small prey through the water column. Their shells, wound in tight geometric spirals and subdivided internally by intricate walls called sutures, provided both buoyancy control and structural strength, allowing these animals to occupy a wide range of depths and to withstand the pressure changes associated with vertical movement in the water column. The diversity of ammonoid shell shapes in the Devonian fossil record, coiled, partially uncoiled, straight, tightly wound, loosely wound, speaks to the ecological variety within this group, each shell shape optimized for a different style of life in a different part of the marine environment. It is within this rich, multi-layered ecological context that we must consider the more subtle and less famous aspects of Devonian vertebrate evolution, the stories of creatures that were not the largest or the most dramatic, but that were, in their own ways, equally important to the overall functioning of the ancient marine ecosystem. Among these, the acanthodians deserve particular attention because they occupy a fascinating and somewhat ambiguous position in the story of early vertebrate evolution, a position that has only recently begun to be clarified by new fossil discoveries and new analytical techniques. The acanthodians, sometimes called spiny sharks, though they were not true sharks, were a group of early jawed fish that were actually among the first jawed vertebrates to appear in the fossil record, predating the great diversification of placoderms and persisting alongside them through the Devonian and into the Carboniferous before eventually going extinct. They were generally small fish. Most species were less than 30 centimeters long, with slender, streamlined bodies covered in tiny, rhomboid scales, and with one particularly distinctive feature that gives the group its informal name: prominent spines preceding each fin, made of bone or dentine, projecting from the leading edge of every major fin on the body. These spines were not simply decorative. They stiffened the fins, making them more rigid and potentially more effective for maneuvering, and they may also have served a defensive function, making an acanthodian more difficult to swallow for a larger predator. The ecological role of acanthodians in Devonian communities appears to have been primarily that of mid-water or near-surface plankton feeders and small prey hunters, occupying the middle tiers of the food web, consuming zooplankton, small invertebrates and juvenile fish, and themselves serving as prey for the larger placoderms and early sharks. Their small size and abundance made them ecologically significant despite their individual inconspicuousness, the kind of animals that rarely dominate paleoecological narratives but whose absence would fundamentally restructure the communities they inhabited. Recent research has suggested that acanthodians may be more closely related to the ancestors of bony fish than previously thought, placing them at a critical node in the early branching of the jawed vertebrate family tree. But this remains an area of active scientific debate, and the precise relationships of acanthodians to other vertebrate groups continues to be refined as new specimens are discovered and new analytical methods are applied to existing ones. Among the most remarkable ecological communities of the entire Devonian period were those of the ancient freshwater lakes and rivers. environments that preserved some of the most complete and informative fossil assemblages known from this period, and that reveal a world of vertebrate diversity as rich and complex as anything in the contemporary marine realm. The freshwater fish faunas of the Devonian, documented in places like the Old Red Sandstone of Scotland and Scandinavia, the Catskill Formation deposits, and the incomparable Gogo Formation of Australia, give us detailed windows into the lives of Devonian vertebrates in non-marine settings. The Gogo Formation deserves special emphasis because it has produced some of the most exquisitely preserved Devonian fish fossils anywhere in the world. Formed in what was once a reef-associated lagoon environment in the late Devonian, the Gogo deposits preserve fish in 3 dimensions, a rarity in the fossil record, where most specimens are flattened by the weight of overlying sediment during the millions of years of burial. The 3-dimensional preservation at Gogo results from a specific taphonomic process in which the fish were rapidly encased in limestone nodules shortly after death, before the weight of overlying sediment could compress them, and the limestone matrix protected the detailed structure of the bones within. When paleontologists dissolve the surrounding limestone with acid, the phosphatized bone within is revealed in stunning anatomical detail, details of internal ear structures, of brain cavities, of gill arch bones, of fin ray articulations that are simply not visible in flattened specimens. The Gogo fauna includes a remarkable variety of species, multiple placoderms representing different ecological roles, early lungfish, lobe-finned fish, and early ray-finned fish, all living together in what was apparently a productive, reef-associated lagoon environment. The lungfish from Gogo are particularly interesting because lungfish, formerly called dipnoi, occupy a unique position in the vertebrate family tree as the closest living relatives of land vertebrates among all surviving fish groups. Modern lungfish, of which only 6 species survive in freshwater habitats on 3 continents, retain functional lungs that allow them to breathe air directly, surfacing periodically to gulp atmospheric oxygen, and some species can survive complete desiccation of their water bodies by burrowing into mud and entering a state of estivation, a form of dormancy, until water returns. These abilities, air breathing and tolerance of dry conditions, are precisely the capabilities that would have been pre-adaptations for the colonization of land, and seeing them in the closest living relatives of tetrapods strongly suggests that the common ancestor of lungfish and tetrapods possessed similar capabilities. Devonian lungfish were more diverse and more widely distributed than their modern counterparts, and their fossils, found on every continent, document a time when the group was ecologically widespread and biologically varied. Some Devonian lungfish retained more fish-like anatomical features than their modern descendants, helping paleontologists reconstruct the path by which the group evolved its remarkable physiological capabilities. Others show specializations for crushing hard food items, thick, crushing tooth plates that could crack mollusk shells and the hard cases of other invertebrates, providing evidence that the ecological role of lungfish in Devonian freshwater communities was not simply that of an air-breathing curiosity, but of an active, effective participant in the trophic structure of those communities. The diversity of Devonian ray-finned fish, the actinopterygians, whose descendants today dominate every aquatic environment on Earth, is often underappreciated in accounts of the period that focus primarily on the more dramatic placoderms and lobe-finned fish. But the Devonian actinopterygians were already establishing the basic body plan and the ecological approach that would eventually prove so extraordinarily successful. Small-scaled, with fins supported by thin bony rays and typically possessing a swim bladder for buoyancy control, the ray-finned fish of the Devonian were laying the groundwork for the most species-rich vertebrate group that has ever existed. Cheropes, one of the earliest well-known ray-finned fish, was found in Devonian deposits and provides a detailed picture of what early actinopterygians looked like. It was a small, active fish, perhaps 30 centimeters long, with large eyes, a large mouth armed with small, sharp teeth, and a body covered in tiny, diamond-shaped scales. Its fin arrangement and body proportions suggest it was a fast, agile swimmer that pursued small prey in the open water, and its dentition indicates it was capable of catching and processing fish and invertebrates of various kinds. Cheropes and its relatives were not dominant in Devonian communities. That ecological status belonged firmly to the placoderms, but they were present, they were diversifying, and they were developing the genetic and anatomical toolkit that would, given time and opportunity, transform their lineage into the most diverse group of vertebrates on Earth. The secret of the ray-finned fish's eventual success lies in their extraordinary developmental flexibility, a capacity to modify their basic body plan in response to new ecological opportunities that no other vertebrate group has matched. The fins of a ray-finned fish, supported by flexible rays that can be individually controlled by attached muscles, allow a degree of fine motor control and maneuverability that lobe-finned fish cannot match. The swim bladder, derived from a lung-like structure in the earliest actinopterygians, can be adjusted to maintain neutral buoyancy at any depth without active swimming effort, freeing up energy for other activities. The scales of ray-finned fish are thin and light compared to the heavy dermal armor of placoderms or the thick scales of some lobe-finned fish, reducing drag and enabling faster, more efficient swimming. Each of these features would, over the course of subsequent geological periods, be elaborated and refined in different ways by different lineages of ray-finned fish, producing the extraordinary diversity of body forms, lifestyles, and ecological roles that characterizes the group today. Returning to the marine realm, we must examine more carefully the ecological dynamics of the Devonian open ocean, the realm beyond the reefs and continental shelves, in the deeper and more open waters that lay between the great landmasses. This environment is less well documented in the fossil record than the shallow shelf environments, because the deep ocean floor is an inhospitable place for fossilization. The sediments there are often dissolved before they can accumulate into preservable rock, but the evidence that does exist suggests that the Devonian open ocean was populated by its own distinctive community of vertebrates and invertebrates, different from the reef community in its composition and its ecological dynamics. In the open ocean, the dominant vertebrates would have been the more streamlined and active swimmers, early sharks pursuing schools of smaller fish, large predatory placoderms ranging into open water from their shelf habitats, and various groups of cephalopods that were effective open water hunters in their own right. The food web of the open ocean was structured differently from that of the reef, less spatially complex, more dependent on the vertical cycling of nutrients between surface waters, rich in photosynthetic plankton, and deeper waters, where organic material accumulated through the rain of dead organisms settling from above. The plankton of the Devonian Ocean, microscopic photosynthetic organisms, small zooplankton that consumed them, and the various larger filter feeders that consumed the zooplankton, represented the base of a food web that ultimately supported all of the larger creatures above it. The productivity of the Devonian open ocean was driven, as it is today, by the availability of nutrients, particularly nitrogen and phosphorus, in the sunlit surface waters where photosynthesis was possible. The supply of these nutrients depended on upwelling currents that brought cold, nutrient-rich deep water to the surface, on river inputs from the continents, and on the recycling of nutrients within the water column by the activities of bacteria and other decomposers. Changes in any of these nutrient supply mechanisms could shift the productivity of the open ocean dramatically, with cascading effects on every level of the food web above. This dependence of marine productivity on nutrient cycling is critically important for understanding what happened in the late Devonian, when the expansion of terrestrial vegetation fundamentally altered the inputs of nutrients and of other chemical compounds from the continents into the ocean. The forests that were spreading across Devonian landmasses were not simply producing nutrients through weathering. They were also consuming carbon dioxide from the atmosphere at a historically unprecedented rate, drawing down atmospheric carbon dioxide concentrations significantly over the course of the late Devonian. This drawdown of carbon dioxide had climatic consequences. Less carbon dioxide in the atmosphere means less greenhouse effect, which means lower global temperatures, and the resulting cooling of the late Devonian climate was a significant environmental stressor for marine communities that had evolved under the warm conditions of the Middle Devonian. The story of how the Devonian world unraveled is one of the most complex and scientifically debated topics in the entire field of paleontology. Unlike the end Cretaceous extinction, which was caused primarily by a single dramatic event, the impact of a large asteroid, the late Devonian crisis was a protracted affair, extending over millions of years and involving multiple overlapping environmental stressors that interacted with each other in ways that are still not fully understood. There was not one late Devonian extinction but several, a series of ecological crises, sometimes called pulses, each of which eliminated significant portions of marine biodiversity, interspersed with intervals of partial recovery before the next pulse struck. The most severe of these extinction pulses are known as the Kellwasser event and the Hangenberg event, named for the geological horizons in which their effects are most clearly preserved in the rock record. The Kellwasser event, occurring near the boundary between the Frasnian and Faymunian stages of the late Devonian, roughly 372 million years ago, was particularly devastating for the reef communities of the Devonian, essentially eliminating the stromatoporoid coral reefs that had been a defining feature of marine ecosystems through much of the period. The Hangenberg event, occurring at the very end of the Devonian approximately 359 million years ago, was equally severe, eliminating a large proportion of the vertebrate diversity that had survived the Kellwasser event and fundamentally restructuring the composition of marine and freshwater communities. What caused these extinction pulses? The current scientific consensus points to a convergence of multiple environmental stressors acting simultaneously, a perfect storm of ecological pressures that overwhelmed the resilience of marine communities that had evolved under very different conditions. Among the most important of these stressors was ocean anoxia, the expansion of oxygen-depleted zones within the ocean that we have already discussed in the context of terrestrial vegetation and eutrophication. The evidence for late Devonian ocean anoxia is compelling and widespread. Black shales, which form in oxygen-depleted bottom waters where organic material is preserved rather than decomposed, are found in rock sequences of this age on multiple continents, indicating that the absence of oxygen was a global phenomenon affecting many marine basins simultaneously. For marine animals, widespread anoxia is catastrophic. Animals cannot survive without oxygen, they suffocate, and the process is rapid. An expansion of anoxic water from the ocean bottom upward into the habitable water column can eliminate entire communities of bottom-dwelling organisms quickly, and if anoxia reaches shallow, coastal environments, the most productive and most biologically diverse marine habitats, the effects can be devastating across the full range of marine ecosystems. The timing of the Kellwasser and Hangenberg anoxic events correlates closely with the major extinction pulses of the late Devonian, strongly suggesting that oxygen depletion was a primary killer. But anoxia alone does not explain the full pattern of extinctions. Other stressors were clearly operating simultaneously. Evidence for significant sea level changes, both rises and falls, is preserved in the sedimentary record of the late Devonian, and sea level changes of sufficient magnitude can eliminate the shallow water shelf habitats on which so much marine life depends. A rapid sea level fall exposes the continental shelves, converting marine habitat to dry land and eliminating the communities that had lived there. A rapid sea level rise floods previously terrestrial areas, but may also destabilize the water chemistry of existing marine environments by mixing waters of different compositions. Either direction of change, if rapid enough, can overwhelm the adaptive capacity of marine communities. Temperature changes also contributed. The cooling that resulted from the drawdown of atmospheric carbon dioxide by the expanding Devonian forests was not uniformly distributed. Different regions of the ocean would have experienced different magnitudes of cooling at different times. But the overall trend toward lower global temperatures was a significant stressor for marine communities that had evolved under warmer conditions. Reef-building organisms are particularly sensitive to temperature change. Modern coral reefs are already threatened by the relatively modest temperature increases of contemporary climate change, and the corals and stromatoporoids of the Devonian would similarly have been stressed by significant cooling. The decline and eventual disappearance of Devonian reefs during the late Devonian extinction pulses is consistent with temperature-driven stress acting on organisms that had evolved within a relatively narrow thermal range. Against this backdrop of environmental deterioration, the fate of individual vertebrate groups becomes a story of differential vulnerability. Some groups hit harder than others, some surviving more intact, some managing to find refugia or to adapt quickly enough to maintain viable populations through the crisis. The placoderms, as we have already noted, suffered the most severely of all the major vertebrate groups. By the end of the Devonian, the placoderms were extinct, entirely and completely gone, with not a single lineage surviving into the subsequent Carboniferous period. This is remarkable. A group that had been the most diverse and ecologically dominant jawed vertebrates on Earth for tens of millions of years, a group that had included the apex predators of every major Devonian marine and freshwater environment, simply ceased to exist. The world moved on without them, carrying only the biological innovations they had pioneered. Why were the placoderms so vulnerable? Several factors likely contributed. Their armor, while effective as a defense under normal conditions, was metabolically expensive to produce and maintain, and it made the animals heavier and less maneuverable than unarmored competitors. In a world of declining productivity, where food was becoming scarcer as reef communities collapsed and ocean anoxia eliminated productive benthic habitats, the energy cost of maintaining heavy armor may have become an unsupportable burden. The placoderms were also in many cases specialists, adapted to specific habitats and ecological conditions that were themselves being destroyed by the late Devonian environmental changes. When the reefs disappeared, the reef-associated placoderms had nowhere to go. When the shallow, productive shelves became anoxic, the bottom-dwelling placoderms were suffocated in place. There may also have been competitive factors. As the environmental conditions deteriorated, the early sharks and bony fish, lighter, more flexible, less tied to specific ecological conditions, may have been able to outcompete the remaining placoderms for the dwindling resources of a stressed ocean. The sharks, in particular, with their cartilaginous skeletons that were lighter than bone and their continuously replaced teeth that were cheaper to maintain than the bony dental plates of placoderms, may have been more energy efficient under conditions of food scarcity, allowing them to persist while placoderms starved. The bony fish, with their swim bladders and their agile fins, could exploit a wider range of water column environments than the heavy-bodied bottom-associated placoderms, giving them access to food resources that placoderms could not reach. The interaction of these factors, environmental stress, habitat loss, competitive pressure, metabolic cost, created a situation in which the placoderms could not maintain viable populations across the global perturbations of the late Devonian. One by one, species that had survived for millions of years flickered out. Communities that had been stable for geological ages collapsed. The armored giants of the Devonian seas, the rulers of an underwater world of extraordinary richness and complexity, vanished from the earth. The fate of the Acanthodians was similarly terminal. They persisted into the Carboniferous but ultimately went extinct, leaving no descendants. The jawless fish, the agnathons that had been the original vertebrates, the lineage from which all jawed fish had ultimately derived, suffered severe losses during the late Devonian as well, though the group did not go entirely extinct at the period's end. The lampreys and hagfish, the two surviving groups of jawless vertebrates, are the remnants of what was once a diverse and varied lineage, and their survival into the modern world is a remarkable testament to the durability of even ancient, generalized body plans under the right conditions. But the groups that came through the late Devonian crisis most successfully, the groups that would define the character of vertebrate life in the subsequent Carboniferous period and all the geological periods that followed, were the Chondrichthyans and the Osteichthyans, the sharks and rays and the bony fish. These groups suffered losses of their own during the late Devonian extinctions. They were not immune to the environmental stresses that were killing off so many other lineages, but they survived with sufficient diversity and ecological flexibility to repopulate the world's waters in the Carboniferous and to diversify into the full range of ecological roles that the disappearing placoderms had vacated. The sharks of the Carboniferous and Permian periods diversified explosively in the ecological space opened by the placoderm's extinction, producing a variety of body forms and ecological types that makes the Devonian shark fauna look modest by comparison. Some developed bizarre, spiral-toothed jaws whose function remains debated by paleontologists. Some became flattened bottom dwellers, ancestral to the modern rays and skates. Some became large, fast, open-water predators in the mode that is most familiar from modern sharks. The cartilaginous skeleton, so long a secondary feature in a world dominated by armored and bony fish, now became the architectural basis of some of the most successful predators in the ocean. The bony fish diversified even more dramatically. The ray-finned actinopterygians, which had been relatively minor components of Devonian communities, seized the opportunity presented by the extinction of the dominant placoderms and began a diversification that would ultimately produce the teleosts, the modern bony fish that today comprise approximately 96% of all living fish species. The teleosts, with their highly modified, protrusible jaws, their thin, flexible scales, their sophisticated hydrodynamics, and their extraordinary variety of body forms and ecological habits, are the ultimate inheritors of the Devonians' vertebrate legacy, the group in which the innovations pioneered during the Age of Fishes were most fully realized and most extensively elaborated. Among the survivors of the late Devonian, the lobe-finned fish occupy the most consequential position in our story, because it was from within this group, from among the survivors of the Devonian crisis, that the first true land vertebrates emerged and established the tetrapod lineage that would eventually populate every terrestrial environment on Earth. We have already discussed the anatomical precursors to this transition, the lobe-finned fish like Eusthenopteran and Panderichthys that document the progressive elaboration of limb-like fins and terrestrially relevant capabilities. Now we must examine the transition itself more closely and consider what the earliest true tetrapods were like and what the world they were entering looked like at the very end of the Devonian. The transition from aquatic lobe-finned fish to terrestrial tetrapod was not the work of the late Devonian extinctions. The basic anatomical groundwork for the transition had been laid during the middle and earlier part of the late Devonian, when the environment was still relatively stable and productive. But the environmental changes of the late Devonian may have provided additional pressure that accelerated or reinforced the transition, by making aquatic environments less hospitable and potentially increasing the relative attractiveness of terrestrial or semi-terrestrial habitats. When the waters became anoxic, when oxygen levels in the ponds and swamps of late Devonian lowlands dropped to levels that could not support obligately aquatic respiration, an animal capable of breathing air directly had an enormous survival advantage. The air-breathing capability that evolved in the lobe-finned fish lineage as an adaptation to episodically oxygen-poor aquatic environments was, under these conditions, a genuine lifesaver. The earliest tetrapods that we know from the fossil record, Acanthostega and Ichthyostega from late Devonian deposits of what is now Greenland, and a small number of other forms from broadly contemporaneous deposits in other parts of the world, were animals in transition. They had 4 limbs with digits, genuine, recognizable limbs, not fins, but their overall anatomy still retained many features of their aquatic ancestors. Acanthostega in particular has been interpreted as largely or entirely aquatic in its habits. It had internal gills alongside its lungs, suggesting that water breathing was still important for it, and the construction of its limbs, while clearly limbs rather than fins, may not have been robust enough to support the animal's full weight on land for extended periods. 8-digit hands and feet suggest that the exact number of digits was not yet standardized. Later tetrapods consistently have 5 digits or fewer, and the reduction to pentadactyly appears to have occurred quickly in the early tetrapod lineage. Ichthyostega, found in the same geological deposits as Acanthostega but apparently somewhat different in its ecological habits, had a more robust skeletal construction that suggests greater capability for supporting its weight on land. Its ribcage was deep and the ribs were broad and overlapping in a way that would have helped support the organs against gravity, a necessary adaptation for any animal spending significant time out of water, because buoyancy in water normally provides this support and it must be replaced by other structural means on land. The ear structures of Ichthyostega were unusual and have been the subject of considerable study. They appear to have been better suited for detecting vibrations through the substrate than for hearing airborne sound waves, which may reflect the specific sensory environment of an animal that spent time on land but remained close to the ground, detecting the footfalls of approaching animals through the solid substrate. The world that these earliest tetrapods were beginning to explore on land was a very different place from the rich, complex terrestrial ecosystems that land vertebrates would eventually inhabit. The late Devonian land surface was dominated by vegetation. The great forests that had expanded during the period were a real, substantial presence by this time, but the terrestrial animal community was still invertebrate-dominated. Insects, arachnids, myriapods, and other arthropods had colonized the land long before the first vertebrates attempted it, and by the late Devonian they had diversified into a respectable variety of ecological roles, but they were all small. There were no large terrestrial predators, no large terrestrial herbivores, no terrestrial animals of significant body size at all. The first tetrapods stepping onto land were entering an ecological landscape in which there was essentially no competition at their body size. The entire large animal ecological space was empty, waiting to be filled. This absence of competition, combined with the presence of abundant invertebrate prey and the expanding forest habitats that provided both shelter and structure, created conditions that were, in the long run, extraordinarily favorable for the diversification of terrestrial vertebrates. Once the basic physiological and anatomical challenges of life on land had been overcome, the support of body weight against gravity, the prevention of desiccation, the extraction of oxygen from air, the reproduction in the absence of water, the terrestrial environment offered an enormous range of ecological opportunities that aquatic environments, already filled with well-established communities, could not match. But those basic challenges were formidable, and overcoming them required modifications to every major organ system in the body. The skeleton had to be strengthened to support weight against gravity rather than against water resistance. The respiratory system had to be modified to extract oxygen efficiently from air rather than water. The reproductive system had to find ways to protect eggs and embryos from desiccation, either by encasing them in shells or by retaining them within the mother's body. The sensory systems had to be recalibrated for an environment in which the medium was air rather than water. Vision becomes more important, hearing changes in character, and the lateral line system that is so useful in water becomes useless in air and must be either abandoned or repurposed. None of these transformations happened instantly. They were the work of millions of years of evolution in the Carboniferous and Devonian, and the early tetrapods of the late Devonian and earliest Carboniferous were animals in the middle of this transformation, capable enough on land to exploit the opportunities it offered, but still carrying many of the physiological legacies of their aquatic ancestors. They were amphibious in the literal sense, animals of two worlds, more comfortable in or near water than far from it, dependent on moist environments for reproduction, and carrying the anatomical marks of their aquatic heritage in every bone and organ. The early Carboniferous world that received these first tentative tetrapods was a transformed environment, one in which the great ecological upheaval of the late Devonian extinctions had created enormous gaps in the community structure of both aquatic and terrestrial ecosystems. The marine realm had lost its placoderms and much of its reef structure. The freshwater communities had been extensively rearranged by the Hangenberg extinction event. And the terrestrial environment, though it had not been directly affected by the marine extinctions in the way that marine organisms had been, was itself changing as climate shifts altered the distribution and character of forests and other vegetation types. In this reordered world, the survivors of the Devonian, the sharks, the bony fish, and the early tetrapods were free to diversify in ways that the ecological dominance of the placoderms had previously constrained. The sharks diversified rapidly in the Carboniferous, producing new body forms and ecological types at a remarkable pace. The ray-finned bony fish began the long diversification that would eventually produce the teleosts, the most species-rich vertebrate group in Earth's history. And the tetrapods, freed from competition with established terrestrial vertebrates that simply did not yet exist, began to explore the terrestrial environment with increasing confidence and increasing anatomical sophistication. The Carboniferous period, sometimes called the Age of Amphibians, in recognition of the dominance of large, diverse amphibian-grade tetrapods during this time, was the direct inheritor of the Devonians' vertebrate legacy. Every amphibian of the Carboniferous, every early reptile that emerged from within the amphibian lineage, every synapsid that gave rise to the mammalian lineage, every diapsid that gave rise to the reptiles and birds, all of them traced their ancestry back through the late Devonian tetrapods, back through the lobe-finned fish of the Devonian seas, back through the jawed vertebrates that had first diversified in the Age of Fishes. It is worth pausing here to consider the magnitude of this inheritance, to truly grasp what the Devonian bequeathed to all subsequent vertebrate life. The jaw, invented in the Devonian, enabled every subsequent predatory vertebrate. The internal bony skeleton, elaborated in the Devonian, provided the structural foundation for every subsequent vertebrate body plan. The paired fins, evolved in the Devonian and transformed by lobe-finned fish into proto-limbs, eventually became the arms and legs and wings of all tetrapods. The air-breathing capability developed in lobe-finned fish was the physiological prerequisite for terrestrial life. The sophisticated sensory systems, the lateral line, the developed eye, the olfactory apparatus, refined in Devonian vertebrates, formed the sensory toolkit from which all subsequent vertebrate sensory evolution would build. Even the immune systems of vertebrates traced their sophistication to adaptations that were being refined in Devonian fish. The adaptive immune system, with its capacity to generate specific antibodies against specific pathogens, appears to have its roots in the jawed vertebrate lineage, meaning that the immunological sophistication that protects every mammal, bird, and reptile from infection is ultimately a legacy of Devonian vertebrate biology. The Devonian was not simply an interesting chapter in the history of fish. It was the period in which the vertebrate lineage found its fundamental design, the design that has proven flexible enough to be elaborated into every vertebrate form that has ever existed, from the Carboniferous amphibians to the Mesozoic dinosaurs to the Cenozoic mammals to the vertebrates alive today. Everything that came after the Devonian was, in a sense, variation on the themes that the Devonian had established. The age of fishes was the age in which the vertebrate theme was composed. All subsequent ages have been playing variations on it. The fossil record that documents this ancient world has been accumulated over more than 2 centuries of scientific investigation, beginning in the early decades of paleontology, when naturalists first recognized that the strange, bony, fish-like shapes found in ancient rocks were the remains of real creatures that had once been alive. The history of Devonian paleontology is itself a rich story, a narrative of discovery and interpretation, of scientific debate and revision, of insights that came suddenly and others that accumulated slowly over decades of patient work. The Devonian fish of Scotland, found in the old red sandstone that underlies much of the northern part of that country, were among the first ancient fish to be systematically studied, and the naturalists who examined them in the 19th century were astonished by what they found. Louis Agassiz, the Swiss-American naturalist who published a landmark study of fossil fish in the 1830s and 1840s, recognized the placoderms as a distinctive group of armored fish, unlike anything living in modern waters, and his careful descriptions and classifications established the foundation of Devonian vertebrate paleontology. Hugh Miller, a Scottish stonemason and self-taught geologist who wrote about the old red sandstone fish in eloquent, accessible prose in the mid-19th century, brought these ancient creatures to a wide public audience, describing them with a combination of scientific accuracy and literary imagination that made the age of fishes tangible and vivid for non-specialist readers. Since those early days, Devonian vertebrate paleontology has been transformed by new discoveries on every continent and by new analytical tools that allow levels of anatomical and phylogenetic investigation that the 19th century pioneers could not have imagined. CT scanning, the use of X-rays to image the internal structure of fossils without destroying them, has revolutionized the study of fossil skulls and braincase anatomy, allowing paleontologists to examine the structure of brain cavities, inner ear regions, and cranial nerve pathways in 3 dimensions without damaging the fossil itself. Synchrotron imaging, which uses high-energy X-rays to resolve details at the cellular level, has revealed the microscopic structure of bone and scale in ancient fish, providing information about growth rates, physiology, and evolutionary relationships that was previously inaccessible. Isotopic analysis of fossilized bone and tooth material has provided information about the diets, migration patterns, and physiological characteristics of Devonian vertebrates, while computer modeling of biomechanics has allowed scientists to test hypotheses about how ancient animals moved, fed, and sensed their environment. One of the most exciting frontiers in Devonian paleontology in recent decades has been the application of phylogenetic analysis, the systematic comparison of anatomical features across multiple species to reconstruct their evolutionary relationships, to the question of how the major vertebrate groups of the Devonian are related to each other and to their modern descendants. The relationships between placoderms, acanthodians, chondrichthyans, and the 2 divisions of bony fish have been debated and revised multiple times as new specimens have been discovered and new analytical frameworks have been applied. One of the most significant recent revisions concerns the phylogenetic position of the acanthodians, those small, spiny fish that we discussed earlier as occupying a puzzling position in vertebrate evolutionary history. For much of the history of paleontology, the acanthodians were treated as a coherent group, the spiny sharks that occupied a position close to the base of the jawed vertebrate family tree, potentially ancestral to or closely related to the ancestors of sharks and bony fish. Recent analyses, however, have suggested that the acanthodians may not be a natural group at all, that what we have been calling acanthodians may actually be a grade of primitive jawed fish, some of which are more closely related to sharks and some of which are more closely related to bony fish, rather than a single coherent lineage with a single evolutionary history. If this interpretation is correct, and it remains a matter of active scientific debate, it changes how we understand the early evolution of jawed vertebrates, blurring the boundaries between lineages that were previously thought to be sharply distinct. The position of placoderms in the vertebrate family tree has also been a source of considerable debate. For most of the 20th century, placoderms were considered a primitive side branch of the jawed vertebrate tree, important as the earliest diverse jawed vertebrates, but not directly ancestral to any living group. This view has been challenged in recent years by analyses suggesting that some aspects of placoderm anatomy, particularly features of their reproductive biology and their jaw structure, may be more directly related to the anatomy of bony fish than previously thought, potentially placing at least some placoderms in a position closer to the bony fish lineage than had been believed. These debates reflect the genuine difficulty of reconstructing evolutionary relationships from fragmentary fossil material, and they are a reminder that our understanding of Devonian vertebrate evolution is still developing, still being refined by new discoveries and new analyses in a process that is far from complete. The geography of Devonian vertebrate discovery is itself instructive about the distribution of these ancient animals across the planet. Major Devonian fish faunas are known from locations that are now distributed across every continent: Scotland and Scandinavia, Poland and the Baltic states, China and Mongolia, Iran and Saudi Arabia, Morocco and South Africa, Australia and Antarctica, Canada and Greenland, and many other localities spread across the globe. This global distribution reflects the cosmopolitan nature of many Devonian vertebrate groups. The fact that animals like Bothriolpes, for example, appear in fossil deposits on virtually every continent suggests that this species or its close relatives were genuinely widespread, capable of dispersing across the marine connections between the major continental landmasses, or adaptable enough to colonize the diverse array of freshwater and marine environments available to them. China, in particular, has emerged in recent decades as one of the most important regions in the world for the study of early vertebrate evolution, with exceptional fossil sites in Yunnan province and elsewhere, producing spectacular specimens of early jawed fish, and even of pre-jaw vertebrates that have fundamentally revised our understanding of the earliest stages of vertebrate evolution. The Xiaoxiang and Kuifengtian faunas of Yunnan have yielded beautifully preserved early fish from the Silurian and early Devonian that document the transition from jawless to jawed vertebrates in greater anatomical detail than was previously available, providing crucial evidence for understanding the origin of jaws, of paired fins, and of the internal skeleton that defines the jawed vertebrate body plan. Australia's Gogo Formation, which we have already discussed in the context of three-dimensional preservation of late Devonian fish, continues to yield remarkable new specimens that expand our knowledge of the diversity and biology of Devonian vertebrates. New species continue to be described from this formation, and new analytical techniques applied to previously known specimens continue to reveal biological details that earlier study had missed. The Gogo fish are particularly valuable because their three-dimensional preservation allows the detailed study of internal anatomy, of brain structure, of inner ear morphology, of gill arch configuration, in a way that is simply not possible with the flattened specimens that make up the majority of the fossil record. The story of Devonian vertebrate evolution is not only told in bone and stone. It is also told in living organisms, in the surviving members of lineages that have their roots in the carry, in their living bodies, anatomical and genetic evidence of their ancient heritage. The coelacanth, Latimeria chalumnae, is perhaps the most famous of these living documents of Devonian biology. Long thought to be extinct, the last coelacanth fossils dated from approximately 65 million years ago, and the group was considered to have vanished along with the dinosaurs. The coelacanth was discovered alive by fishermen off the coast of South Africa in 1938, one of the most dramatic zoological discoveries of the 20th century. Since then, a second population has been found in the waters of Indonesia, confirming that these ancient animals, lobe-finned fish whose basic body plan has not changed dramatically since the Devonian, have been surviving quietly in the deep ocean for hundreds of millions of years, outlasting the placoderms, outlasting the dinosaurs, outlasting every major extinction event in the history of animal life. The coelacanth is not literally unchanged since the Devonian. No living organism can be said to have stopped evolving, because evolution is continuous and inevitable. But the basic organization of its body, the fleshy, lobe-finned limbs, the hollow spine that gives the group its name, the overall proportions and fin arrangement, is recognizable in Devonian coelacanth fossils, making it a genuinely remarkable survivor that allows biologists to study the physiology and behavior of a lobe-finned fish directly, rather than inferring it from bones alone. Studies of the coelacanth's locomotion, particularly the way it moves its paired fins in a pattern that resembles the diagonal gait used by walking tetrapods, have provided insights into the evolution of tetrapod locomotion that fossil anatomy alone could not supply. The lungfish, too, are living survivors of a Devonian lineage, and their biological characteristics, air breathing, estivation, slow metabolic rate, simplified body form, provide direct evidence about the physiological capabilities that the ancestors of land vertebrates may have possessed. Modern lungfish genetics have also proven valuable. The genome of the Australian lungfish, which is enormously large by vertebrate standards and contains vast amounts of non-coding DNA that may represent regulatory sequences important in development, has provided insights into the genetic toolkit that was involved in the evolution of the tetrapod body plan, helping scientists understand which genetic changes were associated with the transition from fins to limbs. Beyond these surviving lobe-finned fish, the legacy of the Devonian is visible in the genetics and development of every living vertebrate. The genes that control the development of the vertebrate skeleton, the Hox genes that establish the basic body axis, the genes that specify the identity of individual vertebrae, ribs, and limb bones, are among the most deeply conserved genes in animal genomes, changing only slowly over hundreds of millions of years of evolution. The fact that these genes are recognizably similar in fish and humans, in sharks and amphibians, in the most primitive living vertebrates and the most derived, this genetic conservation reflects the common ancestry of all vertebrates in the Devonian and pre-Devonian seas, the shared heritage of a body plan that was established during the age of fishes and has been maintained with modification ever since. The developmental biology of vertebrate limbs is particularly illuminating in this context. The same sets of genes that control the development of fins in zebrafish and other modern bony fish also control the development of limbs in mice and humans. The Hox genes that specify which segment of a limb is which, the signaling molecules that establish the front, back, and upper-lower axes of a developing limb, the genes that control digit formation. The fact that the same genetic toolkit operates in fins and in limbs, with differences in the details of how the toolkit is deployed rather than in its basic components, confirms that fins and limbs are modifications of the same fundamental developmental program, a program that traces its origin to the Devonian lobe-finned fish that first elaborated the internal skeleton of the paired fins beyond what their ancestors had possessed. As we move toward the conclusion of our journey through the Age of Fishes, it is appropriate to consider the broader significance of the Devonian in the context of life's overall history on Earth, to step back from the specific details of armored fish and lobe-finned explorers and environmental crises, and view the period from the widest possible perspective. What does the Devonian tell us about how life works, about the principles that govern the evolution of complex biological communities over geological timescales? Several lessons emerge with particular clarity. The first is the importance of evolutionary innovation in driving ecological expansion. The invention of jaws in the early jawed vertebrates was a biological innovation that opened up an entire new suite of ecological possibilities, that transformed vertebrates from passive participants in marine food webs into active participants, from consumers of what happened to drift within reach to pursuers of specific, mobile prey. This transformation had cascading effects throughout the marine ecosystem, driving the evolution of defensive adaptations in prey, restructuring the competitive landscape among predators, and ultimately producing the extraordinary diversity of the Devonian fish fauna. Innovation creates opportunity, and opportunity drives diversification. The second lesson is the vulnerability of ecological specialists to environmental change. The placoderms were magnificent animals, enormously successful, biologically sophisticated, ecologically dominant for tens of millions of years. But their success was predicated on a specific set of environmental conditions that proved, in the long run, impermanent. When those conditions changed, when the reefs declined, when the shallow shelves became anoxic, when the warm, stable, productive seas of the Middle Devonian gave way to the disturbed, cooling, oxygen-depleted waters of the Late Devonian, the placoderms found themselves without the ecological foundation on which their success had depended. Specialization that is brilliant under stable conditions can become lethal under unstable ones. The more generalized, more flexible lineages, the sharks, the bony fish, the lobe-finned explorers of terrestrial margins, survived not because they were better adapted to any specific Devonian environment, but because they were more adaptable to a range of environments. The third lesson is the deep interconnectedness of terrestrial and marine systems, the fact that what happens on land does not stay on land, and vice versa. The expansion of Devonian forests had profound consequences for the marine environment through increased weathering, through carbon drawdown and climate cooling, through eutrophication and ocean anoxia. The marine extinctions of the late Devonian created the ecological opening that allowed terrestrial vertebrate life to diversify in the Carboniferous. Land and sea are not separate domains with separate histories. They are components of a single, interconnected planetary system in which changes in one inevitably propagate into the other, sometimes in ways that are difficult to trace but ultimately unavoidable. The Devonian ended, as we have said, not with a single catastrophic event, but with a prolonged period of environmental instability that erased much of the biological richness accumulated during the period's middle phase. By the time the Carboniferous began, officially defined at approximately 359 million years ago by the Hangenberg extinction event that marks the Devonian's close, the world had lost the majority of its reef-building communities, the entirety of its placoderm fauna, and a significant proportion of the diversity that had characterized Devonian marine ecosystems at their peak. What remained was a reduced, reorganized collection of survivors, sharks, bony fish, early tetrapods, and the various invertebrate groups that had weathered the extinction pulses from which the subsequent history of vertebrate life would be built. The world that those survivors entered was not a wasteland. It was impoverished compared to the Devonian at its height, but it was alive, and it was full of opportunity for the lineages that had come through the crisis. The Carboniferous would see the diversification of the first truly diverse shark fauna, the first explosive radiation of the ray-finned bony fish, and the emergence and diversification of terrestrial vertebrates into the ecological void left by the absence of any prior large-bodied land animals. The groundwork laid in the Devonian, the jaws, the internal skeletons, the limb-like fins, the air-breathing capability, would be built upon, elaborated and refined in the periods that followed, producing over hundreds of millions of additional years, the full diversity of vertebrate life as it exists today. The reefs that built the Devonian world eventually gave way to new reef systems constructed by different organisms. The Carboniferous seas would eventually develop their own distinctive reef communities, dominated by different groups of organisms, expressing a different biological character but serving the same ecological functions as their Devonian predecessors. The principle of reef building, the construction of complex, three-dimensional biological structure by the collective